Okay, here in the wonderful world of photosynthesis, if you go way back to our list of players, you'll see that we had two substrates, H2O and CO2. So I need to give you a little bit of fascinating biology that will tie in to the chemistry that we did way at the beginning of the semester to kind of illustrate where these substrates come from. The phenomenon is kind of the answer to our understanding of what plants are doing around us. I mean, this whole thing is just so amazing to me because all of this carbon that we are, we're carbon-based organisms, all of that carbon started off as CO2 that then got converted into organic carbon, the carbon of organic molecules. CO2 around us, that's the source, that's the reservoir. So the process by which we get these things is called transpiration. And we have to do a little bit with this. There's more to do. We need to look at leaf structure. We're going to look at to understand this. So this is kind of our list of things. I guess I should call this substrates for photosynthesis. H2O, CO2, transpiration as a process, and leaf structure. So this is our outline for what we're going to do in this beautiful little kind of end to this photosynthesis week. Okay. So here's a look at a leaf. If we saw a leaf like this, looks like some kind of an oak leaf. And this is our leaf. We will say that this is the underside of our leaf. Underside. Because this is an oak leaf. What we would see on the bottom of this, of course the whole thing would be green. I don't think I'm going to shade the green right now. But what we would see would be all of these tiny little structures that could stand out. And I'm going to... You wouldn't see these with the naked eye. But they're here. And I'm going to blow those structures up on the bottom to kind of give you an idea what they are. These are called stoma or stomata. Actually, it's the plural, so maybe I should have a couple of them here. Stomata, and these are valve-like openings in the plant. So if we draw, I'm going to draw these. Here's what a stoma looks like. In fact, I'm going to shift to green. That's our color now. Because these are plant cells, and therefore they're completely full of chloroplasts. And then there's other plant epidermal cells around this. So these are regular plant cells of the epidermal plant cells. Plant cells are generally rectangular, as we described a long time ago. They've got cell walls. Okay, so this is my little picture of one of these things here. So these cells are called guard cells. So these cells here are called guard cells. And as I've drawn this thing right now, this is closed. And these things are capable of opening. And so they bow out, kind of become more banana shaped. And when they bow out, they leave an opening. So here again is the kind of surrounding plant epidermis. Just to complete the beauty of the diagram, I'll identify the text pictures. So this here then is space. That's that's open now. So this is our open position, and that gives us the opportunity to identify what's happening. In the open position, we can have CO2 coming in, and we can have water coming out. You'll see that in a minute. And then, of course, very impressively, we can also have O2 coming out. So O2 out, H2O out, and CO2 in. This is actually an exchange of gases then. So this is a gas, this is a gas. The gas exchange. We have a similar problem. We have to exchange gases, but we do the reverse. We go back to last week, and we see that we're bringing in O2 and putting out CO2. These guys are doing the opposite of that. The great balance. So these are called stomata. Stomata is the plural. And they're usually on the undersides of plant leaves. And now I want to give another board that actually shows kind of a section through a leaf so you can see the relationship of these things to the inner structure of the leaf. Okay. Okay, so now we're going to take a look at this leaf. Here's our leaf. Here's our leaf. And what we want to do here is actually slice the leaf. And if you slice this leaf this way and looked at it in kind of a side view, here's what you would see. You'd see all these very long, thin cells on the top. I'll label those in a minute. And then you'll see these really cool kind of plant cells that are pretty tightly packed and kind of like a column. And then I'm going to draw down here some more epidermis. Then boom, here is a guard cell. Here's a guard cell in a closed position. And here's a guard cell, and here's a guard cell in an open position. And then I'm going to have some other cells that are just kind of floating in this space. I'll explain that in a minute. So, again, all of these cells would just be completely full of chloroplasts. I described that later on. So that's what the green is. And we're going to label what these layers are. So this is the upper epidermis. This is the columnar mesophyll. Whoops. Very photosynthetic. This is big time photosynthetic tissue. This is the spongy mesophyll. And then this is the lower epidermis. And then the cool thing about that is that all of this area here, this is space. Obviously, this is a closed stoma, and this is an open stoma here. So, like I showed you before, it's through that open one that we're going to be having this process of CO2 coming in, and as I mentioned, H2O's coming out, and as I mentioned, O2 is coming out. O2 is coming out. So basically, therefore, all of these substances are here to varying degrees. I'm going to describe to you actually what drives this process. It's quite simple. So here's kind of a timeline of what happens in this space. So what we want to do is look at this space through time. What's going to happen through time is that there's going to be a decrease in the concentration of CO2. Why? Because that's being used by the Calivin cycle. So what this is going to do is produce, at this point, we'll have concentration of CO2 outside, out of the plant, will be greater than the concentration of CO2 inside the space. So that's a gradient. Then the other thing is that O2 is being produced in the space, so there's going to be an increase in the concentration of O2 that is basically being generated the O2 that's being generated in the photosystem. Do you remember? Do you remember in photosystem 2 we have the splitting of water? So you have O2 increasing in concentration, and this is from splitting of water. Therefore, we're going to have a gradient here, where the concentration of O2 inside is greater than the concentration of O2 outside. So now at that point, before this is open, O2 is building up inside, wants to go out. CO2 is becoming less abundant inside, so from outside it wants to go in. Those are our opposing forces that once these guard cells open up, that can happen. The rest of the story has to do with water and how water gets here, and this is spectacular as well. Okay. So now, and then I'll just remind you where we are. So this is now a super sketch of my upper epidermis. my spongy mesophil. I just wanted to show you this. That's kind of what we just drew, but this is a little stylized. The thing I wanted to talk about here is that we've got H2O coming in here. Now, H2O, what's going to happen every time that the stomata open up, H2O will escape. It escapes. And the reason it escapes is there's more H2O in the space than there is outside. It escapes to the atmosphere when the stomata open up. Now, here's the crazy thing about O2. You remember all this. You remember that O2 is this spectacular molecule with partial charges. Remember that great stuff? And then remember what water molecules do. So for instance, there's another water molecule right here with its partial charges. And this water molecule here is forming our big friend, the H bond. So now we can set the stage for the big picture. So here's your plant. That's terra firma. Here's the root system of a nice, well-established tree. Beautiful looking plant here. You're familiar with this stuff. And then we're going up. Right. Okay. And then we're going to get some beautiful all right, there's a cool tree. So here's how this is going to work then. The last piece of this is that through all of these plants is an inner system of tubes. And these are these plants that I'm talking about are the higher plants. So this is basically plant xylem. Now, xylem is a vascular tissue in plants. And basically xylem are dead cells. That's wood. You know that the living part of a tree is that outermost part that's just up underneath the bark. But inside, those cells are dead, and they are kind of many of them wood, technically. Xylem is wood, by the way. Xylem equals wood. There's actually two kinds of xylem. There's spring wood and summer. And this is just a kind of spring and summer wood. The spring wood is the wood that if you basically saw a tree down and you have that really smooth kind of view of those growth rings. Those are annual growth rings. The light-colored ones that are thicker, that's spring wood, which is spring xylem. And then the dark ones are summer wood. And you can actually start to count your rings and age grade the tree. You all know that. So what those tissues are for in the tree is a whole bunch of tiny little conduits for the movement of water. So, and by the way, these conduits go all the way up into a leaf. So you remember our little leaf diagram here. This is our little leaf diagram here. And what I did with that leaf diagram is I showed you some little vein structures. And they're branching. You know this. If you ever take a good look at a leaf, actually, let's get our color here, because these xylem tubes are going all the way up into the plant, and therefore down here you'd actually see these tubes, traveling. If you look at one of these structures, you'll see tubes that are in the leaves, and therefore we have an incredible system of tiny little water straws that go all the way from the roots, where H2O then is going in, H2O in, H2O in, H2O in, H2O in. This is how it works. And then water goes up. And you're thinking, well, why does water go up? Water goes up because all these water molecules have their hydrogen fingers in the oxygen belt loops of other molecules. Hydrogen bonding, that's what we talked about before. Water loves itself incredibly. And all of those properties of water, so many of them had to do with water loving itself, being incredibly attracted to itself. And we see therefore this process that anytime when stomata open up, so basically when stomata open, H2O escapes. That's the big deal. So here we have that. It's all traveling up because what's driving this process is H2O escaping. H2O is escaping when the stomata open up because there's more water in those spaces. More water in those spaces than there is H2O. I'm just going to get a little bit more H2O coming out. And then we've got all this water going right to being pulled right to the next station. That's what happens. This extraordinary process is accompanied, as I mentioned, by the escape of O2, or O2's coming out. O2's coming out. I'm running out of space. Whoops, that's not CO2. O2 coming out. And then, let's see. O2 is yellow, so those are CO2s. Doggone it. Okay, I got to change all of that. This is bad news, but not that bad. Okay, so O2 is escaping. O2. 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 That's what's leaving. Breathe some of that in, because that all came out of a plant. Actually, out of plants collectively. So O2's coming out, and then the red was that during that same opening and closing thing, CO2's going in. CO2 is going in. That is insane. I apologize for that. Truly insane. But the idea here, CO2 in, H2O out, and O2 out. That is the big special picture for us. Okay. So that right there gives us the whole big balanced picture of how this stuff is moving in the atmosphere. And then that's great biology. Okay. Okay, so now we're going to put this whole thing together. So we have the light reactions and what they gave us, and now we've got to get into the Calvin cycle. We've got to put all this thing together because we need to make, ultimately, glucose, right? That's the whole thing here. We need to make glucose. So before I can talk about the Calvin cycle, we need to identify the parts. So I guess, start off here. Calvin cycle. We call these the light independent reactions. So these ones do not require light. That's the idea. Light independent reactions. The Calvin cycle. So we'll start off with a where is the Calvin cycle. And this is going to be in the stroma. Of course, it's the stroma of the chloroplast, right? So the stroma of the chloroplast. And then I guess we should say what? What is the Calvin cycle? The Calvin cycle is going to be 10 enzymes that catalyze a series of reactions. 10 enzymes catalyze a series of reactions that yield glyceraldehyde 3-phosphates. Whoops. Aldehyde 3-phosphates. We call these G3Ps, and we've seen them before. So glyceraldehyde 3-phosphates, and we're going to actually use these. So these glyceraldehyde 3-phosphates, these will be used to make glucose and other organic compounds. Okay? So what we need to know now is what goes in. So what goes into this process? First thing is going to be ATP. And of course, as always, let's make it shine. ATP. And ATP is from ATP synthase. In the light reactions. Then we need NADPH. And that is going to be from the light. Actually, by the way, that's dependent. The light dependent reactions. And lastly, we need CO2 from the atmosphere. And this is via gas exchange. In the tissue, in higher plants, actually. I was just going to say via gas exchange. That's enough. There's a whole nice lecture piece that goes with that. So here is our focus going in. ATP, NADPH, CO2. These guys here are going in to a whole big beautiful system of enzymes, 10 enzymes in the stroma. It's called the Calvin cycle, and it is extraordinary. So I'm going to go to the PowerPoint to use a figure from the textbook to describe the general cycle of the general Calvin cycle. But man, you can have a lot of fun. You put a Google on that, and you can get some really, really big, beautiful, impressive kinds of Calvin cycles that show structures of the various substrates and the names of the enzymes. Our textbook doesn't do that. Our textbook keeps it simple. I think that's good for us. So let's go there now. Yay! So here we are. It's time to take a look at the Calvin cycle. So the Calvin cycle, as mentioned before, is in the stroma of the chloroplast. So remember, that's that innermost space that actually also includes the thylakoid discs, but it's that space inside. So I've identified all the parts. We know that we've got ATP and NADPH. Those are coming from the light-dependent reactions, and we know that we have CO2, which is actually coming from an extraordinary process of gas exchange that occurs in plants. So let's start with this great process. Now, as mentioned, if you took a look at this thing in more detail, the kind of detail that a biochemistry course would be looking at, for instance, you would see the names of all sorts of enzymes, a total of 10 of those enzymes. Here, we're basically going to come in with our CO2 molecules. Now, we have cycles. That is a full cycle. And for one cycle, 3 carbon dioxides come in. So what we want to do is kind of get a look at this thing in terms of the numbers of carbons coming in. So I think it's important. What I'm going to do is use a little red pen. So now we have 3 carbons that are coming in here. Notice over here, we're going to bring this in with this here, which is ribulose bisphosphate, I believe is the name. This is cool. You've got 1, 2, 3, 4, 5 carbons. So I'm going to write here 5C. Not only is it a 5C, this is a 5C that has 2 phosphates. And remember before we discussed this process, the B here it stands for bisphosphate, which was 2. So now we have a 5C, and then the other thing is we need to know that we've got 3 of those going in a cycle. One cycle involves 3 of these. So I think what you would want to do is just do a tally of total carbons. So I'll do a little formula here. 3 times 5, we've got a total of 15 carbons right here. So here we have our 3 of the CO2. These are just, this is a 1C molecule, of course. And we count this intermediate, which is 1, 2, 3, 4, 5, 6. So this thing right here is a 6C. And we have 3 of those. So now we're going to basically see that 15 carbons plus, so 3 times 1 is what this is. Times 1 equals 3. And maybe I should try to change my color here. We're basically then going to see that this 15 is going to be equal here. So 3 there. And changing color here to red again. So this would be 6 times 3. So those values equals 18. Now I'm going to get my green again. I'm not very efficient right now with this, but that's okay. Whoops, please. Pen color. Here we go. Okay, so there's 3 total carbons here, 15 total carbons here, and 3 plus 5 should equals 18. So 3 plus 15 should equal 18. 3 plus 15 equals 18. So there you go. That's the kind of thing that I want you to see is the symmetry that we've got our carbon counts always adding up. All right, so I'm going to get back to my red pen and get back to the question in hand. So we've got these arrows, these steps. Here is 3-glycerophosphate. Now we're going to actually be looking at this process here. There's 6 of these because these are 3C molecules. These are 3Cs. 1, 2, 3. So I need to throw right here, there's a splitting event that has taken place here. Notice the split. Boom. There's a split here. So now we have a six total of these 3 carbon entities. And so therefore, if you did that math, we're still going to see the 18. 6 times 3 equals 18. And I think we don't even need to write that. Now we get to the excitement of bringing in ATP. And you can see, you can see here that we're going to bring in 6 ATPs because we have a total of 6 glycerophosphates. And boy, what do you think is going to happen? We're going to take one of those phosphates and phosphorylate this and make a bisphosphoglycerate. Wow, that's awesome. So one phosphate here, this process, that shows that we're using ATP. It's a 1-to-1 ratio, so every single 3-phosphoglycerate plus 1 ATP makes 1, 1,3 bisphosphoglycerate. And that's one of those great symmetrical molecules. And now we're going to bring in this guy, NADPH. So remember we made NADPH, and this of course is a cofactor. This is a high energy electron kind of taxicab as we described it. So this thing is going to deliver its cargo. Now the real idea is that as it goes from NADPH to NADP+, it's giving off electrons, losing electrons. So remember that this was a reduction, which I'm going to just put RED right here to show that this was a reduction reaction that took place. We're going to strip off one of these phosphates. It's weird. We put one on and now we're going to strip one off. And in this whole process, we're going to end up with a total of 6 G3Ps. That is huge because in the cycle we've got 6 G3Ps. G3Ps is what we really are making. That's what ultimately is going to become glucose and other organic molecules. So here we have that G3P. So in this case, I want to go back up here. The NADPH has been oxidized. I did make a mistake, and I am not going to redo this lecture. Sorry about that. It has an electron here, and it gave up an electron, and therefore it became oxidized. So I don't know what I was thinking. Whoops. But the deal is that we have therefore given electrons to the system to reduce. So they're coupled, remember? Redox reactions. Okay, sorry about that. All right, now the cycle continues, but this is the cool thing. Notice, I'm having trouble. Notice that we are going to end up with 5 of the G3Ps staying in the cycle, 1 of the G3Ps coming out as output. And again, if you add up all your carbons, here's a total of 15. If you add up all your carbons here, here's a total of 18. If you add up all your total carbons here, there's 6 of those molecules. 6 times 3 equals 18. So everything stays symmetrical all the way through. Same thing here. 6 3Cs, there's a total of 18. So what we did, all that math just holds. Because with a 15 carries all the way up here. But by the way, we've got to get this back to a bisphosphorylated entity. So that's going to take 3 ATPs. Come on. 3 ATPs, and we end up with, again, this. And now it's a 5-carbon molecule with 2 phosphates. Anyway, you do not need to memorize this, obviously, because you guys are going to be able to crack it open and answer questions. It's in the book. It's an extraordinary process. And the thing that I want to do, and I'll change to maybe some kind of a new color here. Let's go with a blue pen. It's all about this. So every cycle of every cycle of the Calvin cycle gives us one of these. So 2 cycles would give us glucose. Please, please, please. So 2 cycles gives us 2 G3Ps. 2 G3Ps, that's a total of 6 carbons. That's going to ultimately give us glucose. 1 glucose. There we have it. We've got the whole story now. We have put everything together. So we looked in chapter 9 at taking glucose and systematically breaking it down to carbon dioxide and other stuff. And now in chapter 10, we've looked at taking carbon dioxide and ultimately converting it into glucose. That great symmetry, the balance of life, extraordinary. So I'm going to get back on the board. I'm going to finish this up by showing you some exciting things about how in the world this CO2 gets here. That's what we really want to focus on now. So I'm actually going to get an even funkier kind of pen. Let's see. It looks like that's going to have to be magenta. So right up here, what we need to do now is focus on how we get that CO2. How does the plant get it? How it actually got. How does the plant get the CO2? That's what we need to focus on. All right, so that's all I have for the PowerPoint lecture. I'll stick this in, get this thing online, watch this lecture, and have a lot of fun. Bye. Okay, I'm going to give you a list of some of the crazy figures that go with what I just did, which was the process of transpiration and gas exchange in plants. So here's some text figures for plant transpiration related phenomena. So I guess that the first one goes way back to our book. This one actually is relevant. Page 189, and this is figure 10.4. So that's one. And then we've got one on page, this is way back, 769, and this is figure 35.18. All right. Continuing on, there's a couple of good figures. Page 792, figure 36.11. And then finally, page 793, figure 36.12. So there's some good figures if you want to actually sink deeper into this. You can see a little bit more of what I was showing there, but it's an extraordinary process. Again, I can't emphasize enough. Breathe it in, take a good breath, get that O2, put that CO2 out, and then think about where that gas came from and where it's going. Where'd the O2 come from? Where's the CO2 going? Think of it specifically though, in terms of phenomena in organisms. Photosynthesis. Wow. Okay. I think we've done it. We've got a fabulous week. And now we're going to start talking about cells and the division of cells. So, your jobs as always, here's your jobs. Again, watch video lectures. Take good notes. High quality notes, as we always do. Relate these things. So take good quality notes. You also have optional homework. Remember that optional homework. Bring punches. You have, if you want, SI sessions with the fabulous Jenna or others. SI sessions. These are also punches. And of course, do not forget about your quizzes. Stay on this game, you guys. Don't save it. Don't let it build up. Stay on this game. This is really cool stuff. And I can tell you in all honesty that everything, all the way through the end of class, is cool. In the not too near distant future, we're going to be solving genetics problems, inheritance problems, and then we're into DNA and the whole picture. So we've got a lot of great things to do. It's a strange time, but this stuff is awesome no matter what the time. So saddle up and let's have fun.